Hey guys, good to see you again. You're probably enjoying your extra snow day right now. I just got the call that we're going to be off on Tuesday now as well. So I was like, well, no better way to enjoy a snow day than put some video notes together for you guys. So we are going to start talking about genetics at the beginning of the second semester, and it just so happens to be my favorite topic. I'm hoping that you're going to find it interesting. One thing I wanted to bring up that kind of shows you how complicated or how complex genetics can be is Labrador retrievers. A lot of you may have had a yellow lab, chocolate lab, or black lab, as you see in the pictures here. But really, you wouldn't have a yellow lab if there wasn't for some interacting of genes going on, and that is called recessive epistasis. Basically, you have a dominant trait in coat color and a recessive trait in coat color in labs. Black is dominant, chocolate is recessive. Now, you get this masking trait or a masking gene that will actually cover up whatever that color that lab was supposed to be, and that is how you get that yellow lab. So the way you tell that you would have had a chocolate or a black lab is by looking at the mucous membranes of that yellow lab. You can normally notice that the lips, eyelids, and the nose that either be a browner color or a blacker color, depending on if that individual dog would have been a black or chocolate lab. So it's kind of interesting. But let's get into just the introduction and learn some of this new vocabulary. What is inheritance? Number one. Every living thing has a set of characteristics inherited from its parent or parents. And we call genetics the scientific study of heredity or of this inheritance. And as you see in the pictures, you have a family up here in the right-hand corner. They all share some physical characteristics that make them look a lot alike. Same thing with the polar bears and the giraffes. You can obviously tell that this is a baby giraffe and that you have baby polar bears. They look exactly like their parents. So 11.1 talks about the work of Gregor Mendel. He was an Austrian monk. He's basically considered to be the father of genetics. He worked with different true breeding, which means purebred, which means they have their own set of genes. It's not mixed with anything else. You know, I'm sure you've heard of the word mutt before when describing dogs, as they're mixed with a whole bunch of different breeds. A true bred would mean that it's only been mixed with genes of the same dog type, like golden retriever and golden retriever. So he worked with true breeding pea plants. And the reason he chose pea plants, because they're self-pollinating, so you can easily reproduce them. They seed in one season, so it doesn't take very long to see the results of the offspring. And there are also many different true breeding types. Now, some of those different traits that he looked at, there are about 7 of them. We had seed shape and color. There is seed coat and color, pod shape and color, flower position, and then plant height. Now, what is a trait? A trait is a characteristic like eye color. As you can see in the picture here, we got green, blue, browns, and different shades of each of those. Another one could be height. As you see in these two basketball players, you got tall versus short. And then you have your round versus wrinkled pea shape. And those pictures are right there at the bottom of the screen. They're all different types of traits or characteristics that get passed on from one generation to the next by genes. Now, Mendel called the original plants that he started with the parent generation, or P1. And then your offspring, or basically the plants that come from those parents, they're known as the F1 generation. If you cross 2 parents with different traits, the offspring are going to be called hybrids because they're getting a mix. So if we had a tall and short plants, you mix them together, or you bred them, then you would have a hybrid. So this is what Mendel saw from his first F1 cross. The offspring ended up with the characteristics of only one of his parents. So if you look here, this parent can only pass on a capital T representing tall. This parent is short and can only pass on a short allele. So when they come together, you result in this hybrid or mixed version. You got a big T and a little t. So big T is dominant, meaning tall. Little t is recessive, meaning short. So all of the offspring are hybrids. But what you notice is they still represent tall plants, even though they're carrying that short T allele for that short trait. They still are showing up tall because tall is dominant and it is masking that recessive trait. So here again is what he concluded. Inheritance is determined by factors that are passed on from one generation to the next. Those factors are chemical factors that we now call genes. Different forms of the same gene are called alleles. For example, that we just saw the gene for height, there is an allele for a tall plant and an allele for a short plant. This is known as the principle of dominance. Some alleles are dominant and some alleles are recessive, and those dominant alleles are going to mask those recessive alleles. So again, those dominant traits, they're always expressed, and I'm going to use the term always lightly because there are some exceptions to that rule once we get further on into the chapter. But generally speaking, with regular dominance, dominant traits are always going to be expressed. And then you have your recessive traits that are only going to be expressed when the dominant allele is not present. So you're going to have to get that recessive trait or that recessive allele from both parents. All right, now Mendel wondered if the recessive alleles had disappeared or were they still present in the F1 plants, or basically the offspring of the parent generation. So he decided to allow 7 kinds of F1 hybrids, okay, those are offspring from 2 different parents, to produce their own offspring. So they're called the F2 generation because they are the second set of offspring. So here you see that Mendel looked at the results of his F1 and F2 crosses together. So we already looked at the F1 cross earlier, which if you remember produced all tall plants, okay, but they were big T, little t, they were hybrids. Now the F1 offspring are going to become the parents for the next generation, so they are now known as the P2, or the second set of parents. And we have tall plants crossed with tall plants. And when that happened, what Mendel noticed was he actually did get, in the second set of offspring, tall plants and short plants. So what this showed him is that short, or that recessive allele, or that recessive trait showed back up right here. Okay, it was a 1 to 4, about 25% of the offspring were short, and 75% were still tall because that is a dominant trait. Okay, so again, what he reasoned, because that short trait reappeared, the alleles for tallness and shortness had separated from each other when the gametes or sex cells formed. Okay, so remember, sex cells for us are like sperm and egg. For plants, it's a little bit different, but basically this parent, this circle here with both alleles is representing the parent. It's either going to pass on a big T to its offspring or a little t. And so they call this separation segregation, okay, because they are both being passed on separately. Alright, we are going to stop there for this video. The next video I will put up will be over 11.2, which is talking about probability and Punnett squares. And Punnett squares we will use to basically predict what the offspring are going to come out as in a genetic cross. See you soon.